Well, good afternoon. My name is Richard Liebman, the Scientific Director for the National Institute for Biomedical Imaging and Bioengineering, and it is my greatest pleasure today to welcome Professor Robert Langer to the NIH Wednesday afternoon lecture series. Professor Langer has received so many prestigious awards and has had such a vast number of major scientific accomplishments in his distinguished career that it's really an impossible task to provide an adequate introduction in just a minute or so. But I'll just mention a few things. Professor Langer is the David H. Koch Institute professor at MIT and is one of only 14 such professors. He received the highest honor that can be awarded to an MIT faculty member. And his research is at the interface of medicine, cancer in particular, and materials science and chemical engineering with a special focus on nanotechnology, including the development of nanoparticles to treat cancer and other diseases. Dr. Langer is considered a pioneer of many new biotechnologies. His lab designs polymers, lipids, polymer lipid, hybrid nanocarriers for improved drug delivery, as well as controlled delivery systems for genetically engineered therapeutic proteins and DNA and RNA. Dr. Langer's research also includes creation of novel approaches for the engineering of tissues and organs. And he's helped translate his inventions into the clinic by founding more than 20 biotech companies. Dr. Langer has authored more than 1,200 articles and has had more than 800 issued and pending patents worldwide. He's the youngest person in history at the age of 43, one of a very small group ever, to have been elected to all three of the United States national academies, the Institute of Medicine, the National Academy of Science, and the National Academy of Engineering. He's received more than 220 major awards. The highlights of those include the U.S. National Medal of Science, the U.S. Medal of Technology and Innovation, the Charles Stark Draper Prize, which is considered engineering's Nobel Prize, the Millennium Technology Prize, the Albany Medical Center Prize, the Priestley Medal, which is the highest award given by the American Chemical Society, and last year the Wolf Prize in chemistry. This year he's already been awarded the $3 million Breakthrough Prize in the Life Sciences for his research. In 1999, Forbes magazine named Dr. Langer as one of the 25 most important individuals in biotechnology in the world. And again in 2002, Forbes selected Dr. Langer as one of the 15 innovators worldwide who will reinvent the future. In 2001, both Time and CNN named him as one of the 100 most important people in America and one of the top 18 in science or medicine in the United States. Dr. Langer earned his Bachelor of Science degree from Cornell University and an SCD from the Massachusetts Institute of Technology, both in the field of chemical engineering. He holds 20 honorary doctorates from universities in the US and abroad. The title of his lecture today is Biomaterials and Biotechnology, from the discovery of the first angiogenesis inhibitors to the development of controlled drug delivery systems and the foundation of tissue engineering. Dr. Langer.
Thank you very, very much. It's a pleasure to be here. I thought I'd start out, and I had lunch with a number of the young people here, and I thought I'd start out and maybe tell you a little bit about how when I was that age, I ended up getting into sort of the medical field. When I was a graduate student at MIT in the 1970s, I was studying chemical engineering. That's what I got my doctorate in. And that year, 1974, just like a couple of years ago, there was this big gas shortage, and it was actually probably even worse then. Not only did the prices of gas keep going up, but if you lived in Boston, where I lived, you actually had to wait in line at the gas station for about 2 hours to get your tank filled. The consequence of that, though, is that if you were a young chemical engineer, you got a lot of job offers. And pretty much every one of my friends in my class, that's what they did. They went to an oil company. So I thought that's what I should do, too. I interviewed at oil companies, and I actually got 20 job offers. 4 actually from Exxon alone. And it wasn't like I was that great or anything. They just had a lot of job offers, openings. So, but I remember one of them made quite an impact on me. I remember going to Exxon in Baton Rouge, Louisiana, and when I was there, one of the engineers said to me, you know, if you could just increase the yield of this one chemical by 0.1%, he said, that would be wonderful. He said, that would be worth billions of dollars. And I remember flying back to Boston that night thinking to myself that I really didn't want to do that. And I started thinking more about what I did want to do rather than just follow, you know, all my friends. And one of the things I did when I was a graduate student at MIT is I helped start a school for poor children, and I got very involved in creating new chemistry curricula. So one day I actually saw an ad in a chemistry journal to be an assistant professor at City College of New York developing new chemistry curricula. And I thought, that's great, that's what I want to do. So I wrote them a letter applying for the job, but they didn't write me back. But I really liked the idea, so I found all the different universities that had assistant professor to do chemistry education, and I wrote them probably about 40 different ones, and I wrote 40 letters, and actually none of them wrote me back. You know, I guess I wasn't in the right box, you know, the chemistry education box. So that wasn't going real well. So I started to think, how else could I use my chemical engineering background to help people? So I thought about medicine. And so I wrote to a lot of hospitals and medical schools, and they didn't write back either. But one day, one of the postdocs in the lab where I was mentioned to me, he said, Bob, he said, there's a surgeon in Boston and named Judah Folkman. And he said, sometimes he hires unusual people. He actually thought very highly of Dr. Folkman. I won't say what he thought about me. And I wrote to Dr. Folkman, and he was kind enough to offer me a position. In fact, I met Mike Gottesman there. But at any rate, when I went to Dr. Folkman's lab, the project that he asked me to work on actually would be to see if we could actually isolate what would be the first inhibitor of angiogenesis. And I thought I'd start out with a picture, actually in the New York Times in 1971, showing Dr. Folkman's vision of how tumors grow. And it's actually still fairly accurate today. His idea was that somehow tumors cells would become abnormal. They'd grow as a three-dimensional mass to about 1 millimeter cubed, but they might not grow beyond that because they ran into a nutrition problem. Cells in the center would die because they couldn't get nutrients or get rid of waste. But the tumor would solve that problem, he said, by secreting a substance which he called TAF, or tumor angiogenesis factor. And what it did is your surrounding blood vessels normally were quite quiescent. But in the case of the tumor, the blood vessels would begin to grow like crazy right towards the tumor, and they would nourish it and feed it, and the tumor would grow, and also it would metastasize through those blood vessels, eventually killing. And his idea was that if you could stop blood vessels from growing, maybe that would be a new way of thinking about treating cancer. So how do you think about isolating an inhibitor of angiogenesis? We broke this up into 2 parts. One is you have to have a substance that could do this. And one of the things we started thinking about was cartilage. Cartilage is in your nose and your knee, and it doesn't have blood vessels. And there were baby rabbits in the lab, and so myself and Henry Brehm and others would get those, and we got some of their cartilage. But there wasn't, there wasn't very much in the little rabbits. So one of the things you learn as an engineer, chemical engineer, is scale up. So I went to cows, and I went to a slaughterhouse in Cambridge, and I got cow bones, but I still couldn't get that many from them. So I found out where do all the cow bones go in the Northeast, and they went to these meat packing places in South Boston. So I made an arrangement with them to get all their bones, and I'd bring them back to the lab about 3 times a week, and I would scrape off the meat. And I'd get this. This was I'm actually always kind of proud of this. But this is a scapular bone. You just slice this off, and then you extract the cartilage with things like guanidinium hydrochloride and so forth. And then we would put it through various purification procedures so that at the end of a period of time, like a couple of years, I'd maybe have 50 or 100 vials of different substances I wanted to test. That then brings up the second question. How do you test something like this? How do you develop even a quantitative or semi-quantitative assay for something like angiogenesis? And if you look back at the history of medicine, really a lot of factors or substances, the bioassay ends up being rate limiting. How do you think about developing a quantitative assay? Well, Mike Gimbroni had worked in the lab before I did, and he had shown that if you put certain types of tumors in the eye of a rabbit, like V2 carcinomas, blood vessels will grow from the edge of the cornea to the tumor over maybe a 2-month period. So we thought, what if you could take a polymer that A, would be inert in the eye, which isn't easy, and B, could deliver the kind of large molecules in cartilage, which had never been done before, over a 2-month period. Maybe we could develop an assay because we could use an ophthalmic microscope to measure the length of the longest blood vessels and see how fast different substances would grow to the tumor. So that was the idea. Now, the immediate challenge to doing this was developing a controlled release polymer that could meet those two criteria. It turned out Dr. Folkman was on the board, a scientific advisory board of the one company in the world at that time that had done any work on this idea of delivery systems, a company called ALZA. He went out there, they had a very outstanding scientific advisory board, went out there and he asked them, could they help? And they said no. They said, really, you just can't release a large molecule that, if it's over about 200 molecular weight, it's not going to be able to diffuse through the polymer. It's kind of like saying, could any of us walk through that wall? Even the literature at that time would say things like that polymer matrices has been virtually restricted to small molecules. So really the only thing that I had going for me is I didn't know any of that. So I tried anyhow, and I spent about 2 years working in the laboratory, experimenting with different techniques. I actually found over 200 different ways to get this to not work. But eventually, I was able to make these little microspheres. Here's one cut in half. And what we were able to show is that you could release, this was from Nature in 1976, you could release molecules of virtually any size for over 100 days. And later on, using some engineering approaches, we worked out ways to even get constant release. This is albumin being released. In 1976, 2 years after I had started work on this, I actually got asked to give the first talk I ever got asked to give at a scientific meeting. It was actually in a big polymer center, Midland, Michigan. And I was very nervous about this talk. Before this, really, the only talk I'd ever given was in 8th grade. And that didn't go real well. In 8th grade, I had to give this 1.5-minute speech, and so I remember writing it out, and I rehearsed it 3 hours in front of my parents' mirror the night before. And now the day came the next day, and I got up in front of my 8th grade class, and I didn't bring the sheet of paper up, I should have, and I recited it. And I remember, like, for the first 1 minute and 2 seconds, I did all right, and then I could not remember the next word. So I stood up there in front of my 8th grade class, frozen for the next minute, until finally my 8th grade teacher told me to sit down and gave me a not particularly good grade. I think it was an F, actually. So now, many years later, I was very nervous giving this talk or any talk. By the way, I'm going to try to not do that this afternoon to you. So I gave this talk, and this was 38 years ago? My God. Yeah, 38 years ago I gave the talk, and so I was a lot younger. And I was giving it to a very distinguished audience of polymer chemists and engineers. And so I practiced this talk for about 2 weeks. And I remember when I got finished, I thought I did much better. I didn't forget too much what I was going to say. I didn't stammer too much. So I thought when I was done with this talk that these older chemists and engineers, being nice people, would want to encourage me, this young guy. And when I got done, I stepped off the podium, and a whole bunch of them came up to me, and they said, "We don't believe anything you just said." They said, just like what the ALZA people said, that a large molecule couldn't diffuse through solid polymers. And also, we were using organic solvents, and they said that would destroy whatever we would put in. But people, actually, the first people to repeat this, I think, were people at the National Eye Institute, and then many, many more. using it to isolate different growth factors. And so the question would shift in a couple of years to the fact that maybe it wasn't impossible, but how could it work? And I should say that this actually had a profound effect on my career early on. I was telling the people at lunch, when I was a postdoc, actually no chemical engineering department would hire me. And even the department that did hire me originally, with the nutrition and food science department, I remember a year after I joined it, the department head left, and then they had 2 associate department heads, and one day they decided to give me advice. Their advice was that I should start looking for another job. And I also got lots of grants turned down and so forth. So that was like a sort of a rocky start. But anyhow, people began to reproduce it and use it more and more. And then the question shifted to how could it work? So to understand it, what we did is I'd take techniques that were normally used in pathology, like using a cryomicrotome, and I'd cut thin sections through the polymer. So here's a 5-micron thin section of one of the polymers we used, ethylene vinyl acetate. And if you tried to have a molecule diffuse through it, that was over 200 molecular weight, it wouldn't be able to do it. It wouldn't go from one side to the other. But it turns out that if you put a drug in it, here's a red one, and cut a thin section, what you see is, before any release, this phase separation. Polymer here, drug here. If you release this for a year, and then you cut a thin section, what you see is left behind where the drug was are these pores. They weren't there 2 slides ago. These pores are large enough so that molecules, even millions of molecular weight, can get through. But it turns out that these pores have tight constrictions between them. They're interconnected. They're also incredibly winding and tortuous. So it takes a really long time for the molecules to wind their way through. One way I sometimes explain this to people is it's kind of like living in Massachusetts. It's kind of like driving a car through Boston. In chemical engineering, we have this term called tortuosity, and this and Boston have high tortuosity. And actually, over the years, our students have developed models and understanding of how to regulate that tortuosity, so you can actually make systems release anywhere from days to years, or any time in between. Well, now that we've done this work, we could begin to address the angiogenesis problem. Here's the assay that I mentioned. Now we could take all the different fractions and test them. And we probably did 2,000 rabbit eyes. It was an enormous amount of work. And I should say that almost every fraction we tested did not work. And those could be almost viewed as controls. But one fraction did. And I apologize for the blood, but basically here, about 8 weeks after the start of the experiment, you see blood vessels growing from the edge of the cornea over the polymer, which you can no longer see, on their way to the tumor. If you waited about 2 more weeks, this would be out of the 3rd dimension in the eye. Ultimately, it would kill, but we sacrifice them generally at this point. With the inhibitor, in a number of cases, which you never see in any of the controls, the blood vessels did not connect with the tumor. The tumor never grew, and they actually avoided the polymer. So we published this in Science. It was really the first demonstration that you could do in vivo. You find substances that could stop blood vessels from growing. But as people may know, it took another 28 years from that paper in Science before the first angiogenesis inhibitor was approved by the FDA. It took literally the work of many companies and billions of dollars. But starting in 2004, what happened was many angiogenesis inhibitors would be approved, not only for different forms of cancer, but also for eye diseases like macular degeneration. And Avastin, which is mentioned up there, which is a Genentech drug, probably one of the number 1 or 2 biggest selling biotech drugs in history. And now angiogenesis inhibitors have probably been used in over 20 million patients for treating different diseases. It also seemed to me that the polymer technology we developed might be useful not only for the study and isolation of informational macromolecules like angiogenesis inhibitors, but might be useful in their own right, therapeutically, because so many molecules you want to give, maybe by injection or other means, you can't because their half-lives are so short, they're destroyed immediately. And a lot of these large molecules you can't take orally. So just as a proof of principle, Larry Brown, one of my students, put insulin in a little aspirin-sized pellet and showed that you could actually get in vivo release for over 100 days. Dr. Folkman said to me one day, he said, Bob, this was maybe 1976, we're at Children's Hospital, it's a pretty good hospital. But you know, that hospital never had a patent. He said, we should file for a patent. So we did. And the examiner turned it down. In fact, he turned it down 5 years in a row. And finally, the lawyer for the hospital said to me, he said, Bob, this is crazy. He said, the guy's never going to allow it. He doesn't understand it. You should just quit. But I don't like to do that. So I started to think, how could we convince the patent examiner? I mean, legally, of course. And I realized that science wasn't working so well. So I had this idea, you know, that if all these people would tell me that we couldn't do this and stuff, I wondered if anybody ever wrote anything down. So there's something that they had called the Science Citation Index, which means I could take a look at our 1976 Nature paper and see what different people said about it over the years. So I did that, and I actually found a lot of interesting things. It's actually an interesting exercise to see what people say about you. But at any rate, one paper in particular I found to be very useful for this exercise. And this was I did this in around 1982. This was a 1979 paper looking back at our 1976 paper. And these actually, it turned out, were 5 of the top polymer chemists in the world. Here's what they wrote. They're describing this field. They said, generally, the agent to be released is a relatively small molecule with a molecular weight no larger than a few hundred. One would not expect that macromolecules, for example, proteins, could be released by such a technique because of their extremely small permeation rates through polymers. However, Folkman and myself have reported some surprising, so that's a very good word for a patent examiner, results that clearly demonstrate the opposite. So I showed that to the lawyer, and he was very excited. He flew down to Washington, showed it to the examiner, and the examiner said, oh, he said, you know, he said, that's very interesting. He says, I will allow this patent if Dr. Langer can get affidavits from these 5 people that they really wrote that. So I wrote them, and they were nice enough to each write me back that they really did write it. And then we got this very broad patent, which we actually would license to some places. I even got involved, as was mentioned, in starting companies. And today, based on this, there are many different products that are helping people. For example, if there are peptides like leuprolide acetate that are 1,200 molecular weight, you can't swallow them or take them nasally. They just don't get in the body. If you inject them, they're destroyed almost instantaneously. So now they're put in these microspheres, and they will deliver these peptides for originally a month, now up to 4 months. There are a whole bunch of others, too. If somebody is schizophrenic, like if you saw the movie A Beautiful Mind, there are drugs like Risperdal, and it used to be like if you saw that movie, Russell Crowe, he got treated with electric shocks. Now he'd probably get an injection of once of these every few weeks. This is a once a month treatment for alcoholism or narcotic addiction. This is very recently approved system for a once-a-week injection of a glucagon-like peptide. And so these have been used now in many millions of patients to treat these different diseases. Still, all these systems, and there's more to come, but still all these systems, what they do is they're able to control the drug level and reduce side effects. They're able to protect the drug so you can have it have a therapeutic effect in the body. But what they can't do is all these are injected intramuscularly or subcutaneously. They just stay there. So you're not really able to target the drug to specific cells or tissues. And that would be a great thing if you could do it. How could you begin to think about that? Well, these are all microparticles that you're going to inject subcutaneously or intramuscularly. To get them to be taken up cells, we thought, well, you'll need to use nanoparticles. But the problem once you put nanoparticles or any little particle in the bloodstream is you put a drug in it and macrophages are going to eat it. So we had to figure out a way to disguise these so that that wouldn't happen, or at least it wouldn't happen so quickly. So about 20 years ago, I had the idea with some of my postdocs that we would make these little, what we call block copolymers, with polyethylene glycol, which is FDA approved on the outside, which is very water-like. And so the thinking was that maybe the cells would not eat these because it has the water, and it looks like water, which it sees normally. And so maybe they'd circulate around a long time. So that might avoid the macrophage problem to an extent. Still, you have to get it to the cells that you want it to go to. So here, what we did is, and Omid Farrokhzad came to our lab. He's now a professor at Harvard. What we did is not only put the polyethylene glycol on, but we also put a targeting molecule on, which could be an antibody, an aptamer, peptide, or other molecule. And actually, you have to there's a lot of optimization engineering because you have to get just the right density of each of these. I realize I may not have explained this that well, but I was lucky that about 1.5 years ago that Nova, the TV show, they came to our lab and they did some filming, and they explained it an awful lot better than I do, so I'm going to show you what they said. Oops.
He starts with a nanoparticle of anti-cancer drug. That gets encased in a plastic that releases the drug over time. That, in turn, gets a special wrapping that disguises the package as a water molecule to fool the body's immune system. And last but not least, the address where it should be delivered, a key that will only fit the lock of cancer cells.
I should tell you that a lot of the clinicians I work with tell me that it doesn't really blow the cell up quite like that. But like I say, I can't compete with that. They explained it a lot better than I did. And then Omid and I, did it again to try to move these to the clinic. One of the models we've used is after we did some of this work, we start, and I should say that the NCI was very helpful. You know, we got a cancer nanotechnology grant, and I should also say that most of the work we've had funded by NIBIB, and so the NIH has been great on a lot of these to us. So anyway, but here we spun out this company that actually would build a manufacturing plant to make nanoparticles, and we published actually about a little over 1 year ago in Science Translational Medicine the first human studies, and I'll just show a couple highlights. See, normally if you give a drug, here we picked a Taxotere, and we picked something like PSMA that would be overexpressed in the tumor vasculature. So what we did is target that, and if you normally give a drug by itself or in a regular nanoparticle, it goes, this is a log scale, it goes to 0 pretty quickly. But if you put it in the nanoparticle, it'll circulate around for a long time. And what you can see quantitatively is if you compare the same dose in the nanoparticle as not in the nanoparticle, the area under the curve is 250 times greater. So you're pounding the tumor for days. And so it seemed safer. And these are just hints of efficacy. A lot more needs to be done. But these are CAT scans before and 42 days after. Again, before and 42 days after. And now there's multiple clinical trials going on, plus 3 of the largest companies in the world, Amgen, Pfizer, and AstraZeneca, are putting their drugs in these nanoparticles. So we'll see. It's, like I say, very early, but my hope is that these kinds of things will be useful. And we've continued to do related work on this, sometimes with polymers, sometimes with lipids, to deliver drugs like siRNA and mRNA and so forth, and DNA. So this is kind of maybe one nanotechnology example. I'll give you another, too. I remember also about 20 years ago, I was watching this television show on PBS about how they were making chips in the computer industry. And as I was watching it, I thought to myself, boy, wouldn't this be a great way to make a drug delivery system? Now, again, if you spent 30 years of your life working on drug delivery systems, you might think that about any TV show. But what I did, when I saw this, I thought, why couldn't we make a special kind of chip? Not a chip that you'd use in computers, but with some of the same ideas. Could we make different wells and cover them with like a metal, hermetically seal the drug underneath it, so it would be very stable, and what you could do is you could put different wells in, you could put different drugs in, literally have a pharmacy on a chip if the drugs were potent enough, or different doses of the same drug, and so forth. So he had a graduate student, John Santini, and he worked on that. Here he made, this is an early stage chip, 34 wells in the top, 34 on the bottom, and a United States dime. So this is kind of like a short flat chip, but we've made like cylinders, so you can have injectable chips through the trocar, if you want to call them that. They can be bigger or smaller. You can do all kinds of things like it. Let me just show you how they work. So here's a well. It's covered with gold, or it could be a platinum alloy. We've found several of these that are quite biocompatible. And it would stay like this for years. But if you come along and apply just, say, 1 volt by remote control, in nanoseconds, the cover comes off. And when it does, whatever's underneath it comes right out. So here is an example of that from another paper we wrote in Nature, John Santini, my student. And you see we put different amounts of drug in different wells, and they come out at different times. If you wanted to do the pharmacy on a chip idea, there's multiple drugs in the different wells coming out at different times. So over the years, what we did is we moved this from in vitro to small animals to large animals, and recently, then John actually started a company on this too. And what was done actually about a year and a half ago was the first human trials. And this what I'm going to talk about might almost sound like space-age medicine, but let me tell you a little bit about what was done. So in the trial, we actually put the chips in the human body, and then you can communicate with them over a special radio frequency called the Medical Implant Communication Service Band. It's approved by both the FCC and the FDA. Sometimes people ask me, well, but can't somebody tamper with it and change the dose or stuff? I always say that should be the worst problem we have. I mean, we're still trying to get this to work. But what we also did in this case is we have a special computer code that only the patient or doctor can know. So you have to know the right radio frequency, the right code if you want to change things. Also, one of the things that's interesting about this is we set it up so you have a bidirectional communications link between the chip and the receiver. The receiver, by the way, could be something like this, could be a cell phone or something else. And it gives you all kinds of information, like did you take the drug? Did you take the drug, the battery life, and so forth. Now, to the younger people in the audience, that might not seem so important, but I'll just use myself as an example. So my dad died of a heart attack when he was 61, and I've taken a statin drug now for the last 15 years, once a day. But what has happened to me, I don't think I have Alzheimer's yet, but what happens is, I take the drug, but then maybe I walk back to the bathroom about 2 hours later, and I look at the vial and I say to myself, did I take that drug yesterday or today? So I got the idea that what I should do is when I take the drug, I would turn the vial upside down. But then sometimes I walk into the bathroom about 2 hours later and I look at it and I say, but did I turn the vial upside down yesterday or today? So actually having a permanent record of this that you'd always have about when you took the drug and so forth, what time, everything, I think will actually be useful. Let me tell you the trial. It was actually done in Denmark. There were 8 patients. We wanted to pick a situation that we felt A, could have clinical impact, but B, would be a tough test. Again, people usually will tell you why your idea won't work. The biggest reason why people told us this idea wouldn't work is they say you'll get fibrous encapsulation. It'll shut off the release of a substantially sized molecule. So we picked a pretty large molecule, parathyroid hormone. Also, we felt it could have a real clinical impact because 77% actually of the women who take this as injections drop out. And it's not like you could deliver it continuously, like the microspheres. That actually causes worse osteoporosis. It causes bone resorption. So you want to give it in a pulsatile fashion. So this was one we thought we could really make an impact. And what happened is the doctors would do a little office procedure to do the implant. It takes about 20 minutes. All the women felt it was better than injection. They did these surveys. And what happened in the trial is there was less variability, which actually I don't think is important here, but might be important in some cases, than injections. And there's 3 measures of whether you're treating osteoporosis, calcium, and different collagen fragments, and they were the same as the daily injections. Here's some of the clinical data. This is human data. You see parathyroid hormone levels once you pulse it at day 60, 68, 76, and 84, and they're almost superimposable. This is the early chip we did. Actually, if you look at it, there's a titanium case. Here's the chip part. Inside of it, there's a power source like a battery. There's a computer program, and there's electronic components. On this side, you can't even see on the end, we actually imprinted an antenna. That's how you communicate with it. So there's an imprint of an antenna. We're now making much smaller chips, but this was actually done. You can see a couple things. One, you do get some fibrous encapsulation, but it's not significant. I say it's about 1/20th of what a pacemaker gets. And if you look at histology, it's not bad, no inflammatory cells and so forth. We're moving this now in multiple directions. One, we're actually designing 2-year chips. Second thing that's been very interesting is Bill Gates came to see me about 1.5 years ago, and you may know that he and his wife are very interested in birth control, where the woman is empowered to actually control it. So they asked, could we actually make a birth control device that could be implanted, that could actually last for 16 years, that the woman could turn on and off whenever she wanted. Now, that's not a very easy thing to do with any regular birth control implant, but with this, it's actually fairly, I don't want to say straightforward, but it's doable. Because, again, you have this controller. You could make each well last for a month. You could shut it on or off whenever you want. And so we're actually doing that. They've already given us well over $6 million to do this, and hopefully more to really bring this to patients. Finally, what we're looking at is someday, and we've already done part of this, you could envision you could put sensors on these chips. So you could sense signals in the human body and then tell the chip how much drug to deliver. Well, so far what I've done is I've talked to you about how we could take materials that already exist maybe in nature and do things with engineering to make them do things they never did before. But what about the materials themselves? So I was very curious about this when I worked at Children's Hospital and other places. How did materials find their way into medicine? Was it done by chemists or chemical engineers? Actually, as I looked at this, it almost never was. The driving force for bringing materials into medicine almost always were medical doctors who urgently wanted to solve a medical problem. And what they did is they generally go to their house to find some object that would resemble the organ or tissue they want to fix. And then they'd use it for a patient. Let me give you some examples. Actually, the first one was here. Bill Pierce and others. They wanted to make an artificial heart. This was 1967, so they wanted something that had a good flex life. So they picked actually a lady's girdle because it had a good flex life. That's still what is in the artificial heart today. But as you know, you sometimes get clotting when the blood hits the surface of the artificial heart. Clots go to the patient's brain and they get a stroke and they die. Dialysis tubing was sausage casing. Vascular graft is a surgeon going to a clothes store to find something he could sew well with. Breast implants, one was a lubricant, another actually a mattress stuffing. You can probably think of the logic. So one of the things you learn in chemical engineering is what I'll call chemical engineering design. And that is, you know, what I thought, rather than take this approach, take something from your house, why couldn't we ask the question, what do we really want in the material from an engineering standpoint, chemistry standpoint, biology standpoint, and then synthesize it from first principles? So we picked an example. When we started, the only polymer that was FDA approved for implantation that was degradable were the polyester sutures. They degrade like this. They start looking like this, then they get spongy, and then they fall apart, which is okay for some drugs, but if you had a potentially toxic drug like an anti-cancer drug or insulin, it could dump the drug and it could be fatal. So we said, from an engineering standpoint, that's not what we want. What we want is to prevent this dumping potential is surface erosion. Could we make a polymer that does this? How do you do that? So we made this, like I say, an engineering design problem. First thing I should show you is what does a polymer look like? Polymers, by the way, are basically monomers, which could be anything, connected by bonds. So we said, if we want to make something surface erosion, first question we asked is, what should cause the erosion? What should cause the degradation? Should it be enzymes? Should it be water? Should it be something else? Well, actually, all of you have different enzyme levels. The cellular response around an implant will change over time, again changing the enzyme levels. But all of you have excess water. So we said, if we want to make this reproducible for everybody, let's make water the catalyst. Let's build that in to the polymer we design. Then we asked the next question. If we do that, and we want to get surface erosion, we have to make these monomers very hydrophobic, water repellent. Then we ask the next question, what should the bonds be? If we make these very hydrophobic, we need the bonds to break when the water hits it. So then we went to organic chemistry to look at different bond lability. I always say at MIT, I show this slide, that we found a good use for organic chemistry. And we calculated we'd want to go up here to the anhydride bond. So we proposed the idea of hydrophobic polyanhydrides. Now we have to figure out what these R groups were. So now I started talking to some chemists and toxicologists at MIT, notably Mike Marletta, who was a good friend of mine at the time, still is, but now he's head of Scripps. And he was very helpful to us, and we picked monomers that I felt we could make good polymers out of, but that he felt would not be toxic once they broke down. And we came up with this polymer. Very hydrophobic carboxypropane, slightly less hydrophobic sebacic acid, and we synthesized them. And what was exciting is not only did they come close to showing surface erosion, but by changing the ratio from 0% sebacic acid to 79%, you dial in different ratios, you make these last for different lengths of time, going from, say, 2 weeks to many years. So now if you had this, you could begin to think about using these polymers again for different medical applications. And in 1985, Henry Brehm, a young neurosurgeon at Johns Hopkins, came to see me. Henry is now chief of neurosurgery there today and Cushing professor, and he was looking for a better way to treat glioblastoma multiforme, which is a uniformly fatal disease. Mean lifespan less than a year. The drug at that time, the only drug that was really used, was BCNU. And what Henry and I started talking about was maybe we could have a slightly different paradigm. Rather than give a drug like this systemically, like IV, and have it travel throughout the whole body, maybe we could give it locally. Maybe we could allow the neurosurgeon like Henry to operate on the patient and line, before he closes the patient's brain up, line the surgical cavity with little wafers containing this. Now, this drug normally lasts for 12 minutes, half-life. But if you put it in the polymer, it's protected. It lasts as long as the polymer lasts. And what they wanted was a polymer that would last for a month, and we could do that. They also wanted it to be surface eroding so it wouldn't dump the drug. And the polymer protects the BCNU from degradation, but also you get this benefit: high concentrations in the brain where you want it to be, and low concentrations in the rest of the body where it causes harm. Now, of course, if you're a young professor and you have ideas like this, you have to raise money, and we would write grants to the NIH, NSF, other places, and we didn't do very well. By the way, just to make it clear, when we write grants to NIH, it's actually professors at other universities who review them, not people at NIH. So you have these study sections of professors from other universities. And when we have, and this would be true at NSF too, and here are just some of the reviews that came back. In 1981, people said, the chemists actually said, you could never synthesize these polymers. But I had a very good graduate student at the time named Howie Rosen. Howie later became president of the Alza Corporation, it's a $12 billion company that was sold to Johnson & Johnson. And also Howie's a member of the National Academy of Engineering, and he synthesized them. Sent it back, and the reviewers, people said, we'd still never get this to work because we have anhydride bonds, and the polymers will react with any drug you put in, because anhydrides are very reactive. But we didn't think that was true because they were in a polymer form, and I had another couple postdocs, Bob Linhart, who is a Constellation Professor of Chemistry at RPI, and Cam Leung, who's the James Duke Professor of Bioengineering at Duke University, also a member of National Academy of Engineering, and they showed that there was no reaction. Sent it back again. The reviewer said that the polymers are low molecular weight and fragile, but I had another couple postdocs, Avi Dome, who's now became chair of medicinal chemistry at Hebrew University, and Edith Mathiowicz, who's a full professor of bioengineering at Brown University, and they made polymers high molecular weight, and they were fine. The next issue was that the polymer would be toxic. I had another graduate student, Cato Lorenzen. Cato later became Dean of Medicine at the University of Connecticut, actually a member of both the Institute of Medicine and National Academy of Engineering, and he showed that the polymers, which is what Marletta predicted, would be quite safe. Anyhow, this kept going on until 1996 when the FDA approved it. It's the first time in over 20 years the FDA approved a new treatment for brain cancer, and the first time they ever approved this idea of localized polymer chemotherapy. You can probably tell from the way I'm speaking that I'm very proud of how well all the MIT graduate students and postdocs did. They became endowed chair professors, member of National Academy of Engineering, chair people of major corporations, whereas the reviewers at the other universities, they didn't do so well. I should say, though, and I as an aside, that, in spite of this, I still think that the NIH review process is the best one of any of the agencies because you do get feedback. And unfortunately, and we're certainly going to see this now, is when money is tight, what happens is reviewers usually look to things, they become more critical. They look at why things can't work. And I think it just makes the whole process much more difficult rather than, giving people a chance to dream and do great things. So it's unfortunate when money becomes tighter and tighter. I do want to show you what this operation looks like, but if anybody's squeamish and doesn't like the sight of blood, you shouldn't look. So here are a little wafer going into the brain. Usually they put 6 or 7 in and close up the brain. It's very hard to get good advice when you give a talk, but about 12 years ago, I remember giving a lecture to a group of chemical engineers at MIT, and my wife came to the lecture. And at the end of the lecture, I said to her, Laura, what did you think? And she said, well, Bob, she said, the talk was all right. That, by the way, is incredibly high praise. But she said, I don't know if you were looking at the chemical engineers when you were showing those slides, but a lot of them were turning green and looking at the floor. So ever after that, I did what I did today. I gave a warning, showed them really quickly. But I do want to tell you the sequel to this, which is that 2 years after that, I got asked to give this dinner speech. But this was to a group of neurosurgeons. And at the end of the speech, a number of the neurosurgeons came up to me and they said, those 2 bloody slides you showed? I said, yes. They said, no problem. They said, you could have left those on as long as you want. They said, but those chemical formulas, you know, to leave those on for a neurosurgeon, I guess, is really bad. So anyhow, this was some of the phase 3 data in Europe originally, and you do see a prolongation of life. It's not a cure by any means, but you do see a prolongation of life. And this is now still used 18 years after its approval in about 50 countries. It's again, fairly narrow situation where people will use it in fairly localized tumors. But it's still been used probably in nearly 100,000 patients. Also, the principle of localized polymer chemotherapy is widely used. The place where it's most widely used actually is not in cancer, but in interventional cardiology. In fact, some of this was done by another one of my students, Elazar Edelman, who's now a professor at Harvard and MIT, and as well as a number of companies. And the whole idea is that if somebody has heart disease, you often put in a stent, like a Chinese finger puzzle to keep the blood vessel open, but about 50% of the time it closes off because of smooth muscle proliferation. So now what you do is you take a different anti-cancer drug, in this case Taxol, coat it with the polymer, and it locally delivers it, and these are used in about 1 million patients every year. Last thing I want to talk about briefly today is the idea of using materials not just to deliver drugs, but to deliver cells and create new tissues and organs. And I'll just be brief about it, but it's certainly an exciting area, and I'll just sort of show you a little bit. Here's a patient of one of my colleagues, Jay Vacanti. This little boy is dying of liver failure, and there's really no way for him to be treated except somebody else has to die and they do a transplant. That's what Jay does. He came to see me, it must have been over 30 years ago, and we started talking about this to see if we could come up with a better way to do it. And the idea that we came up with, which is shown from a paper we wrote in Science, is here. We wrote this before people talked about stem cells. But the idea is you could isolate different cell types. If you inject them at random, not much happens. But if you take these same cells and put them close enough together, they have the ability to form structures. People have shown that before us. I mean, at Berkeley, people have shown that you could take mammary epithelial cells, put them close enough together, and they can actually form acini and make milk. Our thought was that we could create a polymer scaffold that would be degradable to allow the reorganization to occur, but then the polymer would go away. Also, what's critical, and I probably won't go into that too much now, is the way you grow the cells. It's more than just regular tissue culture. Sometimes you have to apply the right stresses, and there's a lot of bioengineering in that. And then you could make the tissue. Again, we'd synthesize new polymers. We might use lactic glycolic acid copolymer, but in some cases, certain cells will not adhere to it. So we actually created new polymers like polylactic acid colysine, where through this amino group you could link different amino acids, sequences like RGD or so forth, so we could control what the cells do. We would then form them into different structures like fibers like this. And we've even gone further, like Prasad Shastri, who was one of my postdocs, now runs a big institute in Germany, Polymers. So you could begin to use CAD-CAM techniques like computer-aided design to make shapes in any shape you want. And just to speculate with you that 30 years from now, maybe somebody will go to a plastic surgeon and they say, I'd like a new nose. And my speculation is you'll have a computer screen, it'll have pictures of about 30 different noses. You can take your pick. To that end, he's used the computer-aided design along with various approaches like 3D printing or different methods of making foams. And here's like a regular nose. And you can even put cartilage cells on it. Now, this is, like I say, a regular nose, but let's say somebody wanted an upturned nose. It wouldn't be that hard to do. You just take a little bit of this off. Let's say somebody wanted a hook nose. I mean, they probably wouldn't. But if they did, we'd just give them a little bit more. Again, take the cells and make this. And I'll give you an example of that in just a minute. So I thought I would just give you 2 examples to end the talk, but there are probably 20 or so that we're working on. First is cartilage. We've worked on this with Chuck Vacanti, Jay's brother, and here you could take nude mice, and we've made a scaffold and actually redid this guy's skull. We redid this guy's cheek. If you open the animals and look at them, what you see is you get this pure white glistening cartilage, and actually, interestingly, I didn't bring it, but if you look at histology, it looks identical to textbook cartilage, but it's not. turns out that the levels of glycosaminoglycans and type 2 collagen is less, and the mechanical properties of this are not as good. So we're not at a point where we can help people with athletic injuries. But there are a lot of cosmetic things you could do. And in fact, we have a big project with Jay and Linda Griffith, who's, like I say, my former postdoc, with the Army, to see if we can make different body parts for soldiers who might return from Afghanistan or Iraq, and one of them's like an ear. And to that end, Linda actually made a scaffold in the form of a human ear. Here's a high-powered scanning electron micrograph of the polymer fibers. Here are the chondrocytes multiplying. They're making extracellular matrix. Over time, the polymer will fully dissolve, and you'll get all cells in extracellular matrix. Chuck actually put this on a rabbit, and we have not put it on patients yet, but what has happened is Jay and others have done clinical trials, and I'll just give you one example. Here's a 12-year-old boy at the time, but if you look at him, he's got no chest covering his heart. But like other 12-year-old boys, he likes to play baseball. But you can imagine if he ever got hit in the chest with a baseball, he could die. So Jay operated on him. We made a polymer scaffold for him, took his own cells, and made him a new chest, and he's now 6 feet tall and doing very well. And then the last example I thought I'd give you is actually one where we licensed something to company, but they actually now can make new skin for burn victims who have, or for patients with diabetic skin ulcers. And these are now FDA approved and fairly widely used. Let me just show you a picture of that. So here's a 2-year-old boy who's very, very badly burned. But you can take the product now, which are neonatal skin fibroblasts, put it on the polymer. You can even cryopreserve these. And you can put it on the child at the time of the injury. Looks like this. If you come back 3 weeks later, he looks like this. And if you come back 6 months later, he looks like this. And as I mentioned, these are now FDA approved for both diabetic skin ulcers and burn victims. And I think I will end here. We're working on a whole bunch of other tissues and organs, some funded by NIH. And I guess I would just sort of make 2 comments in closing. One, it's funny, I get asked to give a lot of lectures, but when NIH asks, I feel very, very grateful to NIH over the years. They've been our #1 funding source, and none of these things would have happened, none of these products would have happened if they didn't get their genesis here. But the second thing I'd say is this is really just the tip of the iceberg. I really believe that bioengineering is still in its infancy. I think that the kinds of things I've talked about, we've probably raised more questions than we've answered. And I really believe that as we move forward into this century, there's just going to be so many things that can be done where by combining engineering and biology and medicine, that all kinds of things will be done by the students and postdocs of the future that will relieve suffering and prolong life. Thank you very, very much for having me here. We can turn that off.
Thank you very much for a wonderful talk. Questions?
Sure.
This is the latter part of your presentation of tissue engineering. And now, most of the examples you have shown or we have read in the newspapers, they're more structural. They are the structure of the skin structure. Can you imagine in the future that we will be able to impregnate them with biological molecules so they have enzymatic activities or they have hormone release activities, real biological activities of a tissue?
Oh, could we build biological activity into these materials?
Yes.
Sure. Oh, actually, absolutely. Sometimes we do what we call biomimetic materials so that you could build them. And in fact, one of the things I alluded to, I didn't really go into it much, is the fact that you could make polymers and then take specific amino acid sequences to recreate surfaces that actually aid adherence. But you can also bring in other features too, if you want to, absolutely.
Let's take a tough example of creating neurons. Can that happen? Functional neurons.
Well, I guess the question there, and I'm probably not the best expert on that, but I guess the question there is, could one take stem cells and differentiate them into neurons? I think people have done things along those lines. The question is whether you could, how far you want to go.
No, I meant by tissue engineering, not by going to stem cells and regenerating the neurons.
Right, regenerating, I see. But I mean, I guess I take a broad definition of tissue engineering. In other words, if one, I mean, in fact, we've actually looked at things like in spinal cord repair and others. You could, I mean, I would, you know, what you want to do, at least from a medical standpoint, is if I can get the neurons from stem cells, if I can get neuronal stem cells, that's one of the things we've looked at, I'm perfectly happy to use those. You'd have to still find the right material to do the kinds of things you want to do it with. To do what you're saying, I mean, my guess is somebody is probably smart enough to do it sometime if they get enough funding and they're smart enough, but probably it's not us.
There you go.
Thank you, a wonderful talk. Can you give us any lessons learned or perhaps tips on how to do better translational science to business to commercialization?
Yeah, well, I'm not sure I'm an expert on that, but I guess, so there's a couple different things in there. For us, I think the environment at MIT has been very good in terms of translation. I mean, MIT, I think is, you know, to me, if you're going to raise the types of dollars it takes to, and get the kind of organization it takes to make products, I mean, you need a lot of money. I mean, that's, so therefore you have to have an incentive, create an incentive for people to want to invest in it, and whether that be companies or investors like venture capital. So I think the first thing you need is, first you need a really good idea. That is important. Secondly, I think you need a really good patent. I don't think in the biological area that, you know, the medical area, that people will invest unless they see that you have really good intellectual property. Third, you probably also need good in vivo data, and you also probably want to get yourself associated with really good business people. But that I would think in terms of commercialization. In terms of translation, I mean, that may not be required. I mean, if you work with good clinicians, that also, you know, and we've been fortunate, again, having Children's Hospital and Mass General, and I also have done a lot with Johns Hopkins, and certainly you have things here. I think there's, you know, more and more, I think with biomedical engineering, there's good collaborations between bioengineers and clinicians, going back both ways. So we have, like in our own lab, my lab, I have about 10 clinicians. One of the nice things about these places is you have these fellows who come for 3 years, you know, and they're fantastic. And so we have fellows from all kinds of disciplines, you know, gastroenterology, neurosurgery, dermatology, ophthalmology, you know, pain. So I think I think that translation really requires the interface between scientists and engineers on the one hand and clinicians who really want to make things happen on the other. Sure.
During your talk, you commented on how it's difficult for NIH to review research proposals and still end up choosing the innovative ones. Could you comment on that further, what you might see we should do?
Well, I don't know that it's NIH. You know, I think the sad thing, what I watch, and I was on a study section for 6 years, so I could speak from personal experience. I mean, you know, so what I've seen, and it's first, the study sections have, you know, you could do it one of two ways. You could kind of use what I'd call a benevolent dictator approach, meaning somebody who's in charge who has vision and can just decide what they want to do. And, you know, sometimes there are places like DARPA, and I don't know that they necessarily do that right either, but sometimes the program manager can just sort of do that. I actually prefer what NIH does. I like to have peer review. I like the fact that people sit around a room and you really get a scientific review. So I think that part is good. I think, but I'll give a couple of suggestions. Sometimes I know even today I get a call from people at NIH and they say, well, could you be on a study section 2 weeks from now or 3 weeks from now? And it's sort of hard to do that in such a short time. You know, so I think that you lose a lot of reviewers, you know, along those lines by making things so quick. But the biggest problem to me is the money. You know, what I've watched, when I was involved in these things and things are funded up to the 30th percentile, people will sit around the room and they'll take more chances. I mean, this is the professors at the universities. When things are funded to the 7th or 10th percentile, you know, people want, there's just a natural human tendency to say what can go wrong. And boy, if you're doing something speculative, a lot can go wrong. So I think that, to me, it's sort of the nature of the beast, that when the pay lines are really low, the conservatism, at least in my experience, has become greater. When the pay lines are higher, I think all kinds of good things get funded. So to me, the way it would be fixed is the government give you a lot more money. And then you could give it to us. But I really believe that. I mean, like I say, I'm saying it from personal experience. I mean, I've been involved in those study sections. In other words, you guys probably aren't, but I mean, you might see them. But you know, you sit there for 3 days and you see what the different members say, and some of the comments, I mean, what I showed you on mine was nothing. I mean, you know, it's like you see some of these comments, people get ultra conservative. I mean, when money's tight, that's just the most natural human tendency. And I would add, what's more important than funding the kind of stuff that you and we all do? I mean, that's super high priority. So I just think that that when the pay lines are really tight, I mean, that just makes it very, very hard. And people are human. Other questions?
Maybe I can just ask
sure.
a very quick question then before we...
Sure.
So going forward, do you see limits to the complexity of tissues and organs that could be engineered in the lab?
Sure.
Because you have all these different cell types and
yeah.
putting them in the right place.
Well, I think you're absolutely right. I mean, certainly there's huge issues as you increase complexity. You know, liver has at least 5 cells, and so all kinds of interesting structures, heart. Yeah, I think there's no question that things get more and more complex. So right now, I think we're at the stage where we try to pick things that are easy, maybe one cell type, and maybe doing some structural thing like skin or cartilage maybe, but and even that's very, very hard. So I think, you know, we're at this, and if one does a liver, you know, maybe one tries to just do one cell type that might cure an enzyme deficiency disease or something else. So I think there's no question that the complexity is huge. Nonetheless, if we go, and I wouldn't know where to stop, 50, 100 years into the future, people will probably figure these things out, you know, but you take small steps, you make advances, people will make greater understanding in the biology area and the engineering area, they'll put them together and they'll build on the things that are done. So that's what I meant when I ended the lecture, that I think we're still in the infancy. So I think if we had the same lecture 100 years from now, the kinds of things that you said, there will be very significant advances on, but it will come from not only the kinds of things I talked about, but, you know, fundamental advances in cell biology and engineering as well.
Continuing on this topic, what do you think, I mean, given the complexity of the body, given all the different tissues that need to interact, should we play on the ability of the body to heal itself in terms of, for example, if there is a chronic disease and inflammation, we take care very specifically about directing inflammation, not necessarily suppressing it, but directing it into the way that will allow the body to rebuild itself with some help maybe from outside, but not completely relying on building everything from scratch. So what do you think about this?
Well, I agree with that. Even when we do some materials, you know, some of the ways that we induce vascularization is some materials will create, you know, a vascular response just by implanting them there. So, but, you know, the way I look at it, and I was maybe trying to say that before, I'll take any help I can get. You know, these things are really hard problems, and so materials can do things like bring in blood vessels, and so if you can have some type of natural response that helps you, like you're saying, you know, I think that's a good thing.
Well, I'd like to, first of all, let me tell you that there'll be a reception in the library, and everybody is invited to meet there for a further chat with the speaker. And I'd like to thank our speaker again for giving us such a wonderful and thought-provoking and elegant lecture to us.
Thank you.